All right, so welcome to the virtual edition of Bio 106F. So I'll start with some updates just on kind of the class, and I won't spend a whole lot of time on coronavirus since I already sent an email update with some information on that and some useful links to other information that are, updated in real time. So again, feel free to reach out to me if you have any questions about that. Given the amount of, kind of false information out there, I'm happy to clarify anything you have questions on. But again, the email contains a good amount of information, so just make sure you've read that. In terms of this class, we're going to be doing these pre-recorded online lectures this week, and if we like it, we can keep it. I'll send a poll out after Thursday's lecture so that you guys can kind of weigh in on whether you think this is going to be useful and effective, or whether there's something that you'd, any ideas you have for something you'd rather have me do. This is my first time using this software, so you can kind of let me know, whether that worked well, things of that nature. In terms of labs, we're going to keep most of them. We're going to either do them virtually or any of them that are difficult to kind of do virtually, I'll just replace them with another assignment. And so the lab points breakdown on the syllabus will remain the same. And so your quizzes won't be weighted more heavily or anything like that. In terms of office hours, I'll just be doing them virtually, or you can make an appointment with me. In either case, just let me know, send me an email beforehand so that, I'm not just kind of like sitting on a webcam for an hour just waiting for someone to show up and nobody does, something of that sort. And then in terms of the pass-fail option, I'm sure you guys have been getting a lot of emails about this. This is a new policy that the university decided on. Feel free to reach out with any questions or, if you know for sure you want to do this. Probably you'll want to wait a bit, to get closer to seeing what your final grade is to decide whether you want to do this. I think it's a bit unfortunate that the university chose a C as the cutoff between pass-fail, but it's what they've decided on. My guess is that that's going to be the final policy, and there's not much that I can do about that. But yeah, again, feel free to reach out with any questions that you have about that. Also, I've posted a frequently asked questions from the university to Moodle. I don't know if you guys got that, so feel free to take a look if you haven't seen that. Okay, so for the Unit 3 material, we'll be diving into evolution. And so for this lecture, we're going to start with Unit 2 wrap-up. We're going to start with some biotechnology techniques that I think will be just kind of useful for you guys to know about in your lives, and also help you to write your Unit 2 reflective writing. You won't need to know this for the Unit 3 quiz, but I think it's just useful to know in general. And then for evolution, we're going to start today with some evidence of evolution, just kind of that we can see in the real world. And then in terms of the antibiotic resistance case study, we're actually going to push that to Thursday. So this is going to take the place of the antibiotic resistance lab. But for today's class, instead, we're going to do a sort of natural selection virtual study. It's just a YouTube video, and there will be a question sheet to go with that. Okay, so let's dive into some modern biotech. So we've already discussed CRISPR in our previous lectures, which is a technique used for specific DNA targeting and editing, and of course comes naturally from the adaptive immune function that it plays in bacteria and archaea. We'll also discuss stem cells and some of their therapeutic uses, and then we'll finish up with in vitro fertilization and some kind of derivative techniques of that. So to start off, just a quick CRISPR recap. So we've talked about how CRISPR is this adaptive immune system basically in bacteria in order to allow them to cleave invading phages or plasmids or other foreign DNA. And so CRISPR has been adapted basically by biotechnologists in order to use this engineered Cas9 enzyme that relies on a single guide RNA. And so this creates a simple two-component system for genomic editing. And so basically the user designs the guide RNA sequence in order to target the desired DNA sequence, and then Cas9 can cleave that DNA, and you can have a number of different edits that result from that, or it can be engineered to do other things at that genomic locus as well. including actually some targeted epigenetic engineering, which could allow you to basically target gene expression at a particular genomic locus, which is pretty cool. So you could modify gene expression even using this technique. And so, of course, the first documented use of this in human cell culture was in 2013. And then a couple years ago, unfortunately, we had documented use of this in human embryos, which was, as we discussed, a bit ill-advised for a number of reasons. So that's CRISPR, and obviously this is a pretty exciting future tool for genomic engineering in terms of maybe treating genetic diseases, actually curing genetic diseases within embryos, and maybe potential for applications for gene expression and other uses. So really exciting technology there, just not quite developed to the point that we can use it clinically quite yet, at least in terms of embryo engineering. So stem cells, probably everybody has heard of stem cells before. And stem cells are basically just cells that have not yet differentiated, or they haven't chosen the professional cell type that they are going to be. So there are multiple types of stem cells. The first is embryonic stem cells. And so these are, as an embryo develops, of course, the cells are just proliferating and they haven't really decided what kind of cells they're going to be. And so that's the source of embryonic stem cells, is actual human embryos. And so there are a number of ethical concerns with that, because basically to harvest embryonic stem cells in any significant quantity, you have to destroy the embryo. There are also non-embryonic or adult stem cells. And so in all of us, we have stem cells within, for example, our bone marrow or our spinal cord. And so, even as we have basically all of our cells differentiated, we still have to have these progenitor cells, right, in our bodies to replace other cells that are going to die or, senesce over time after they've divided enough times. And so we constantly have to have this source of basically pluripotent stem cells that can differentiate into multiple cell types to replace cells that we lose. We can also actually create pluripotent stem cells from basically pretty much any type of adult cell. These are called induced pluripotent stem cells, or iPSCs. And so this is another application of biotechnology that's been developed in recent years. And so basically you can just treat adult cells with a cocktail of different proteins and stimuli that tell them to revert into this state where they can differentiate into multiple cell types. And so that's really cool. And then we can also harvest stem cells from cord blood or amniotic fluid during pregnancy. So what's so great about stem cells? What is their potential therapeutic purposes? So stem cells actually have the potential, or may have the potential, to treat or even cure a lot of diseases. So some things that stem cells are actually currently used for in clinical applications: we can use stem cells from bone marrow, peripheral blood, or cord blood, right? So these are places that we can harvest it from adults. And then we can also harvest a patient's own stem cells from either their bone marrow or their spinal cord, or from a donor. And so you guys have probably heard of bone marrow transplants for treating diseases like leukemia. And so the reason that we harvest bone marrow is that it's rich in these stem cells. And so by taking that and transplanting it into somebody with a disease like leukemia, we can replace some of the cells that they're losing and, potentially treat or cure that disease. And then stem cells can also be used to reconstitute immune function following things like radiation or chemotherapy. So chemotherapy, basically its function is to kill rapidly dividing cells. And unfortunately, among those cells are immune cells. And so in order to replace those stem cells from, like, bone marrow, for example, can be used to put some of those back. So some of the potential applications for stem cells that haven't yet gone into practice. Potentially, stem cells can be used to treat injuries or wounds. And so this can be of particular interest to athletes or like burn victims or, others with serious injuries. And potentially, someday maybe we can even use stem cells if we find ways to get enough of them and they're found to be efficacious enough to even treat just like surface level, like cuts, scrapes, things of that sort. Stem cells also have the potential to treat degenerative diseases such as Parkinson's or Alzheimer's. And so since these diseases involve basically just rapidly losing certain populations of cells like neurons or your motor function, things like that, potentially we can use stem cells to basically put those cells back and replace them as they're degenerating and treat these diseases over time or potentially cure them. And then potentially we can also use stem cells, of course, to treat immunosuppression. So we talked about this following like radiation or chemotherapy. But hopefully we could also use this in the future to treat diseases like severe combined immunodeficiency, or maybe immunodeficiencies that result from like viral infection from like HIV or AIDS, things of that sort. And then there are also many other potential applications for stem cells. So where do stem cells come from, and what kind of hurdles and ethics and safety do we have to consider? So for embryonic stem cells, of course, these are in limited supplies because, again, they're harvested directly from embryos. And then there's the question of whether it's at all ethical to use them for treatment. And so a lot of pro-life people are going to take issue with using embryonic stem cells to treat disease because, again, they come from a destroyed embryo. And some might view this as sort of an incentive, to kill, to basically destroy embryos and use them for therapeutic purposes. And so the question is, whether that potential life that would have resulted from the embryo is basically worth taking in order to treat diseases in living humans. So in terms of adult stem cells, some of the hurdles in terms of ethics and safety, we have to think about matching of donors to recipients, just like in blood donations, but this can actually be even more difficult with stem cells to make the correct matches. And so, of course, with we talked about with blood typing, how this can cause pretty severe immune reactions if you put the wrong, blood from the wrong donor into a recipient. And the same is true for stem cells. If you don't properly match the donor to the recipient, you're going to cause a lot of immune reactions and basically problems downstream of that. There's also the question of cost and difficulty of extraction and efficacy, and etc. So basically, is the potential use for them, is it worth the cost, and is it worth the toll on either the person that you're going to have to extract them from or the donor that you're going to extract them from, and the price of all this, proliferating the stem cells, all of these different things, injecting them locally, and making sure they're going to kind of take and serve the right function. All of these are considerations in treating the disease. So the question is, in most cases, whether the disease is severe enough to actually warrant this. And then in terms of the induced pluripotent stem cells, the ones that we can just make from adult cells, there's a question of genetic privacy issues for donors. And so donors might want to be anonymous, if you're going to donate your cells and have them turned into induced pluripotent stem cells. But unfortunately, they're always going to be genetically tractable, right? So the cells that you implant from a donor into a recipient are still going to have the donor's genetic material. So you can always basically trace that back, which is unfortunate in some ways. And then iPS cells are also not entirely the same as embryonic stem cells. So they have different epigenetic landscapes, and in some cases they have less plasticity. So they're not going to be able to differentiate into just kind of any cell type you want. So they have a little bit more restrictions than embryonic stem cells. They also tend to die more upon transplantation and maybe not take as well. And so there are definite reasons, that we're still using embryonic stem cells over induced pluripotent stem cells, despite the perhaps more ethical concerns involving embryonic stem cells. So some of the future challenges and questions for embryonic stem cells and other stem cells is, one, can we stimulate a patient's normal stem cell population to proliferate? And how can we do that? And so basically with just kind of drugs or, different local treatments, could we encourage a patient's own normal stem cell population to continue dividing and producing the types of cells that they'll need? Another question is, what is the optimal epigenetic landscape of embryonic stem cells? And what triggers them to basically differentiate and decide on different cell fates? and how could we recapitulate these without actually harvesting the stem cells? So, are there drug combinations that we could do to basically properly recapitulate the epigenetic landscape to have cells that that aren't necessarily embryonic stem cells, but basically fully function like embryonic stem cells? And then another question is, can we give them the right combination of proteins and things like that in order to have them differentiate into whatever cell type we want them to be? And another question is, what's the best way to deliver stem cells to particular anatomical sites, and then how to keep them there? And so these are all kinds of basically future challenges and questions for this field that'll help stem cells to go into these other potential therapeutic applications. Okay, so now let's talk IVF, or in vitro fertilization. So in vitro fertilization is used to treat infertility that results from a number of health conditions. And so another thing that you can do with IVF is you can actually do pre-implantation genetic diagnosis. And so this can be used to decrease the likelihood of genetic diseases being passed on to the child. And so in some cases, if parents are likely to have certain genetic diseases, basically this can be screened using IVF before the embryo actually is implanted. And so you can screen for embryos that are not going to have these genetic diseases, which is pretty neat. Another thing that you can do with IVF is that you can actually prevent paternal transmission of HIV. So basically by taking the sperm out and kind of washing it first, you can actually wash the viral particles away from the sperm and then use healthy sperm to fertilize the egg. So there are some risks associated with IVF. For example, a lot of the embryos are just not going to survive. I mean, you're kind of taking them out of their normal habitat, trying to grow them outside of a human body, and then reimplant them. So there's a lot of risks with that. And this, of course, raises ethical concerns, right? Especially, again, for pro-life people. The question is, should we be taking these egg cells out at all? Should we be selecting specific embryos? Basically, condemning specific embryos, even if they have genetic conditions. Is it right to just kind of, throw them out, condemn them to die type of thing? And then additionally, up to 73% of IVF embryos are partially aneuploid. So we talked about aneuploidies, right? Like the abnormal amounts of certain chromosomes. And so there are definitely some health concerns related with that. And it's kind of crazy that actually up to 73% of these have these issues. But it seems that since in a lot of cases it's only a subpopulation of the cells that have these, that the resulting embryo and child seem to be viable and for the most part fine. So the process of in vitro fertilization, it starts with basically you stimulate eggs to be produced, then you retrieve them. You then fertilize them outside of the human body, so basically just kind of like in a petri dish or a cell culture dish. And so you can basically fertilize them using a population of sperm, or there's also a technique where you take one particular sperm and just directly inject it into the embryo. You then watch the embryo develop, right, and monitor it, make sure it's going to grow. And at that point, you can do your pre-implantation genetic testing by taking a subpopulation of those cells. And then at that point, you can either preserve these embryos cryogenically, or you can go ahead and implant it into the uterus in order to allow the embryo to grow within then either the mother or a surrogate mother. And so that's the basics of IVF. Now let's talk some kind of derivatives of this technology. So one is called somatic cell nuclear transfer. And so this can be used for cloning. And there are basically two types of cloning that this is used for. One is therapeutic cloning, and the other is reproductive cloning. So how this works is similar to IVF. You're going to obtain a donor ovum cell, and so this is just like a precursor egg cell. You then remove its nucleus, and this is called deprogramming. So you deprogram the cell by taking its nucleus out. You can then obtain a somatic cell, so just like a differentiated cell, from whatever individual that you want to clone. And you insert the whole somatic cell actually into the empty ovum, the egg cell. And so the somatic cell nucleus will then be reprogrammed by the host egg cell. And so it'll actually serve the function then of an egg cell with the donor's genetic information. You can then stimulate the ovum to divide, and it's now a viable embryo. And so this is really cool. So some potential uses for this are in therapeutic cloning. And so a therapeutic cloning is basically one potential alternative for embryonic stem cells. So you could obtain pluripotent stem cells from the cloned embryo, right, that matches the donor's genetic information, and then you have donor cells that are, of course, genetically matched to the recipient. And so you can make, in this way, patient-specific pluripotent stem cells to be used for therapy. This could also potentially be used to generate tissues or organs from the stem cells from transplantation. So you give them the stem cells the right signals right after you take them out, and then tell them basically to turn into a specific organ type, and you could theoretically transplant that into the individual from their own biological information. So some pretty cool options there. This can also be used for reproductive cloning, which is the basis for cloning animals and, theoretically humans. So proof of principle for this in mammals was shown in 1996 with Dolly the sheep. Maybe you guys have heard of that. You can look it up if not. Basically, they cloned, an adult human sheep and had a genetically matched identical sheep offspring that resulted from that. Some problems with this technique: there are high fetal and neonatal death rates, and the offspring will also often have health issues. So the question is whether this is possible at all, given the high death rates and things like that. And people aren't really convinced that if you did this in a human embryo that any would survive to term. So it may not be possible for humans, and even if it were, it's not, there's a lot of questions about the ethics of this, of cloning an adult human. So that's how SCNT works. So now we can talk about mitochondrial replacement therapy, which is another kind of derivative of IVF. And mitochondrial replacement therapy, or MRT, is used to prevent or ameliorate inherited mitochondrial diseases. So mitochondria are a maternally transmitted organelle, and while mitochondrial disease is rare, it is very serious. So MRT is actually not approved for use in the U.S. due to ethical and practical concerns with basically every technique used to achieve this. However, it has been successfully used at least once, which resulted in a child with technically 3 biological parents, right? So how this works is that you take donor egg cells with the mother's nucleus, and then you'll actually implant that nucleus into an enucleated egg cell. So similar to IVF when you took the nucleus out, basically you take the nucleus out of an egg cell from a donor that has healthy mitochondria, and you implant the biological mother's nucleus into that cell, so that the child will genetically match the mother, but its mitochondria will not be the same as its mother's. And then the resulting cell from that can be fertilized by the father's sperm in order to result in a child with the mother and father's nuclear material, but also a third parent is in the mix, which is the donor mother's mitochondria. So that's super interesting, and also, has some ethical concerns of its own. Okay, so that's it for modern biotech techniques. So you don't need to know these again for the Unit 3 quiz, but I'm going to ask you about them, to think about them and write about them for the Unit 2 reflective writing. So just check Moodle once you're done with this lecture for that writing prompt. Okay, so now getting into Unit 3, which is evolution. So today's question is, what evidence do we have of evolution generally and in today's world? So we're going to look at some examples of evidence of evolution in today's world, including human skin color, these Galapagos finches, also called Darwin's finches. We're going to talk about just kind of an intro to Charles Darwin. I'm sure you've all heard of Charles Darwin and his theory of natural selection. And then we're going to talk about some ways that we can actually observe evolution in real time today, including microbial evolution. Okay, so for human skin color exhibits, and we talked about inheritance, right? So this is an example of a trait that exhibits both polygenic inheritance and incomplete dominance. So the color of our skin is based on the amount of melanin that our cells produce. So there are at least 3 genes for melanin production, and each of these genes has 2 alleles. And so for simplicity, we're just going to call the genes A, B, and C. And the alleles, we're going to have capital A and little a, capital B and little b, and capital C and little c. And so basically the combination of these alleles results in more than 7 basic shades of human skin color. So you can imagine if you had all dominant traits in in all of the genes, then you have very dark color. If you have basically 1 dominant and 1 recessive of each, then you're going to have an intermediate color. And very light color results from recessive traits of all of these genes. And so if you were to do a Punnett square of these 3 genes and the resulting traits, then we've done hybrid crosses and we've done dihybrid crosses, and this is actually a trihybrid cross, right? So you can see that here we have 2 parents, each are hybrids for each of these traits, and when we cross them, of course, any combination of the 3 traits can result. And so what we end up with is this 64 square Punnett square, and we can see all of basically the offspring that can result from this cross. And so the offspring of this cross can exhibit up to 7 shades of skin color based on the number of dark skin alleles in each of their genotypes. So what results from this phenotypically are these different offspring with these different colors of skin, right? And so basically you could have offspring from this cross with significantly darker skin color than either of their parents. So at the top of this chart, it shows the number of dark skin alleles that each offspring will inherit. So going back to the table, you can see, like in this case, right, every trait here is recessive. And so in the bottom right corner here, so there are going to be 0 basically dark skin alleles here. And as we go, across the table to the left, we can see we get up to 6 in the top left corner, up to 6 dark skin alleles. And so this is how this basically plays out, and the number of offspring that you can expect from this cross out of 64. So on either pole of the spectrum, right, we're going to have 1 expected with the lightest skin color and 1 expected with the basically darkest melanin color. And then more in the middle, you're going to expect a lot more offspring that are kind of intermediate. And so this is what results from not only the polygenic inheritance here, but also from the incomplete dominance of these traits. So you can actually get a mixture because these traits are incompletely dominant. And so when you have, like, for example, capital A, lowercase a, you're going to have that intermediate melanin production rather than just all-out melanin production from the capital A allele. So that's the basics of the genetics of human skin color. And so now we can watch a little video on how evolution has actually influenced human skin color. And if the volume or anything is weird on this, just go ahead and pull it up on YouTube on your own. You can just pause this lecture, right, and pull it up on YouTube. But hopefully this will work.
Human brains are gray. Human blood is red. Our bones are off-white. Doesn't matter where you're born or to whom. But human skin is different. Some of us have rich, dark brown skin. Some of us have pinkish white skin. Most of us are somewhere in between. For the longest time, why this variation exists was a real scientific mystery. That opened the door for some to invest this biological trait with moral value, and then use that to justify the suffering of others. But biological traits aren't good or bad. They're features that have evolved because they enhance an organism's odds of surviving and passing on its genes.
Like other animal traits, the sepia rainbow of human skin color evolved through natural selection. Now, thanks to advances in anthropology and genetics, exactly how and why it did is no longer a mystery. Biological anthropologists like myself spend our lives studying how humans evolved and why we differ from one another physically. Our skin provides one of the most visible markers of human variability. It's something that sets us apart from our closest animal relatives. Under their dark fur, chimpanzees have pale skin. And millions of years ago, that was probably also the case for the primates that were our common ancestors. So where did humanity's range of skin colors come from? From physics, we know that the color of any object comes from the wavelengths of light that it reflects back to an observer's eye. We see leaves as green because they reflect back the wavelengths our eyes see as green. Absorbing the wavelengths we see as other colors like blue or red. In humans, different wavelengths of light are reflected or absorbed by a pigment in the top layer of our skin. That pigment's called melanin. It sits inside what look like tiny grains, the melanosomes, that are produced by cells called melanocytes. Our individual genetic inheritance determines the type of melanin inside our melanosomes. The reddish-yellow pheomelanin is more abundant in lightly pigmented people. More darkly pigmented people have more of the brown-black eumelanin. And the more eumelanin, the darker the skin. Melanin also colors human and animal hair and the feathers of many birds. Interestingly, the wavelengths of light that melanin reflects are far less important biologically than the ones it actually absorbs. And of the ones it absorbs, the ones that are the most important are those that we can't even see. Much of the light given off by the sun is invisible to our eyes. Some of that is what's called ultraviolet radiation, which is highly energetic. So much so, it can actually penetrate living cells. When it does, it can wreak havoc within them. It can even cause mutations in skin cell DNA. What stands between us and that threat is the melanin in our skin.
Melanin is kind of like the sensor and kind of like a guardian molecule, and its main job is protection.
For instance, it protects skin cell DNA by forming what are called supranuclear caps and absorbing UV.
They're like little parasols around the nucleus, and UV cannot penetrate these to go and attack the DNA.
That's just one of the things molecular biologist Zalfa Abdelmalek finds remarkable about melanin. Another is the broad range of benefits it provides a diverse collection of species.
We know that melanin in lower vertebrates is important for regulating body temperature. It can also give animals camouflage and allows them to recognize other members of the species to propagate the species.
In humans, one of melanin's functions is clearly to protect cells from UV damage. As we evolved, we lost hair and increased melanin production in our skin. So, is there a connection between the intensity of UV radiation and skin color?
Hi, Tess.
I first became fascinated with UV and skin color in the 1990s. But as I searched for information about the global distribution of solar UV, I discovered the available data was in fact quite spotty. I began casting a wider net, and almost by accident found exactly the raw data needed to fix that. it hadn't been collected by anyone interested in my questions, but rather by NASA.
Ignition and liftoff.
In the 1980s, concern about the health risk posed by the depletion of UV-blocking atmospheric ozone led NASA to take millions of UV measurements from space. I asked NASA to send me the data, and then asked my geographer husband, George Chaplin, to try to visualize it. It turned out to be a bigger request than I'd realized, but he found a way to turn all those data points into a map that showed for the very first time exactly how UV exposure varies throughout the world.
So this is the map.
Most striking was the clear gradient between the equator and the poles, which was interrupted only in places where altitude increased UV exposure.
This is actually in the Tibetan Plateau.
And persistent cloud cover decreased it.
Congo Basin, so it's full of humidity and moisture, which is blocking the UV.
Solar energy is a fundamental attribute of any environment, and it's a well-established fact that organisms living at different latitudes adapt in some way to their local solar conditions. To see how closely human skin color correlates with UV exposure, I collected skin pigment measurements made by anthropologists studying indigenous peoples. For many years, anthropologists have faced the challenge of how to accurately measure skin color. We now use this little device called a reflectometer. Basically, it sends out light of specific colors and then it measures the amount of light that is reflected back. This tells us what color Tess's skin is, and we can then compare this to people all over the world. George then created a second map using measured skin colors and environmental data. It showed UV intensity does indeed predict skin color. Wherever UV is strong, skin is dark, like it is near the equator or at high altitude. At the poles, the skin of indigenous people is almost always lighter. That suggests that variation in human skin melanin production arose as different populations adapted biologically to different solar conditions around the world. As we've noted, our early ancestors probably had full body hair covering pale skin, just like other primates. So when did the darker shades of human skin begin to evolve? DNA sequencing has made it possible to find evidence that can help answer that question. Rick Kittles is a geneticist who's skilled at deciphering such clues.
Whenever a species undergoes some form of selection, some form of natural selection, evidence of that selection is found in the genome. And so as geneticists, we get really excited when we explore the genome for these signatures. One way in which that's done is by sampling worldwide populations and looking throughout the genome at variation and comparing across populations. And it's a very exciting process. I feel like a detective when I go through that process.
One of the many genes that genetic detectives have linked to human pigmentation is called MC1R. Sampling from around the world indicates there's a fair amount of variation in the DNA sequence of that gene, but not from every corner of the globe.
When we look at MC1R within African populations, we don't see a lot of diversity. And the particular allele that they have in those African populations is the one that codes for darker skin. MC1R codes for a protein which is involved in the switch from the production of pheomelanin to eumelanin. And we know pheomelanin is the reddish-yellow pigment, and then the eumelanin is the brown-black pigment.
The absence of MC1R diversity in African populations indicates that, in that part of the world, there is strong negative selection against any alleles that would alter dark skin. And how long has this allele been fixed in African populations? Other genetic studies have calculated that it has been as much as 1.2 million years. Since our species evolved in equatorial Africa, it's reasonable to conclude that by that time, all humans were dark-skinned. The fossil record supports what we've gleaned from genetic evidence. But here's where we confront what was, for me, the heart of the mystery. The evolution of dark skin in humans suggests that under strong UV light, that trait provided a survival advantage. So what exactly was that advantage? It's certainly true UV damage to skin cell DNA can lead to cancer, and skin cancer can be fatal. For a long time, that seemed the likeliest explanation. Except, skin cancer generally develops after a person's peak reproductive years. For that reason, though it might cut your life short, it's unlikely to affect your ability to pass on your genes. As I was struggling to conceive of an alternative explanation, I happened to attend a lecture on severe birth defects. That talk was about a research project that had found evidence that certain birth defects are far more common among pregnant women with diets deficient in a B vitamin called folate. Only weeks before, I'd come across a paper that described how strong sunlight breaks down folate circulating in skin blood vessels. Here was a direct link between UV radiation, skin color, and reproductive success. It was a small eureka moment for me. In the years since, we've learned that folate is not only essential for normal embryonic development, it's even needed for healthy sperm production in males. Folate is biological gold. It is an essential nutrient, and it needs to be protected from UV radiation as it circulates in the blood vessels in the skin. That is what melanin does. I felt I was halfway home on my quest to understand human skin color variation. But a big question remained. Why aren't we all dark-skinned? It turns out there's another side to our relationship with UV light. UV light is not all bad. In fact, the small portion of it known as UVB is critical for the synthesis in our bodies of vitamin D, a process that starts in the skin. Without vitamin D, humans cannot absorb calcium from our diet to build our bones and for a healthy immune system. Back when all our ancestors lived close to the equator, there was no problem getting enough UVB through dark skin to make the vitamin D needed. But then some populations started moving north, where the UV striking Earth's surface is much weaker. In northern latitudes, dark skin makes it hard to produce the vitamin D that human bodies really need. The consequences of vitamin D deficiency include rickets, a bone development disease that can cripple the young. In higher latitudes with less UV, the selective pressure on MC1R that produced dark skin in our ancient ancestors began to abate.
When we look at the early movement out of Africa, when that constraint was relaxed, we then see a plethora of variation.
In European and Asian populations, geneticists have discovered greater variation in the MC1R gene, but less variation in several other genes, ones associated with lighter skin types.
Different environments led to other genes being selected for and being important for those populations in terms of skin color.
Selection for light skin gene variants occurred multiple times in different groups around the world, some of it in just the last 10,000 years. Support for the idea that the UV vitamin D connection helped drive the evolution of paler skin comes from the fact that indigenous peoples with diets rich in this essential vitamin have dark pigmentation. The tension between these two aspects of our biological inheritance, On the one hand, the need to protect ourselves from most ultraviolet radiation, and on the other, the need to use some ultraviolet radiation for our own benefit. These forces drove the evolution of the wonderful variation in human skin color that we see around us today. It's the legacy.
Okay, so just to recap, human populations that have spent more evolutionary time further from the equator tend to have lighter skin. And this human color distribution map also reflects the amount of UV light that reaches each of these places. So, in the video they mentioned that humidity can also help to block some of these UV rays. And so, in places where you have kind of high humidity, like rainforestish places, you don't necessarily have as much melanin production as in places where there's more arid climates close to the equator. So what evolutionary benefits do the different amounts of melanin provide? So higher melanin helps to protect from skin cancer. However, skin cancer tends to occur later in life after the reproductive years. So this isn't necessarily a trait that natural selection would act very heavily upon. So to be healthy and have healthy offspring, individuals do need adequate amounts of particular vitamins, and some of these vitamins can be affected by UV light. So in particular, these are folate and vitamin D. And so strong UV radiation destroys folate. And so in places where there's strong UV radiation, in order to keep the required amount of folate, more melanin is basically advantageous. However, there's a fine balance here because UV light is also necessary to produce vitamin D in the skin. And so given that the balance between these vitamins is crucial, evolution has actually selected for optimal melanin production for this balance relative to geographical location. So this process of selection is known as natural selection. And natural selection is a theory that was proposed by Charles Darwin in his book On the Origin of Species in 1859. And so again, I'm sure you've all heard of Charles Darwin, probably Darwinism, and there are some sort of misconceptions about Darwin and his theories. And so natural selection is actually simply the principle by which each slight variation of a trait, if useful, is preserved. And so it's not always simple as, like predator-prey relations, or everyone that has a disadvantageous trait is going to die off. The principles of natural selection are actually that, one, variation exists within populations of organisms. And so we know that this is due to genetics and alleles of genes, right? Darwin actually knew nothing about genetics. So he observed that variation existed, but he really didn't know how exactly it was created. And he also knew about inheritance, but not genetic inheritance. So people knew about, again, if children tend to look like their parents, organisms clearly inherit traits from their parents, but it wasn't clear why back then. So the second part of natural selection is that there is a struggle for existence due to limited resources. So there are going to be more individuals that are going to be born than will survive. Individuals then with favorable adaptations are most likely to survive and produce offspring, and therefore favorable adaptations are inherited and passed on to subsequent generations. And over long periods of time, successful variations produce differences that result in the formation of new species. And so this is the process of evolution by natural selection. And again, on the topic of the origin of species. And so this is how we can get speciation in new species from existing ones based on these slight variations, basically. And so Darwinian fitness is this concept of the ability to produce offspring over generations. And so Darwinian fitness can be thought of pretty simply as just kind of the ability to get your genetic information into the next generation by all means possible. And so, when we talk about survival of the fittest, I think what comes to mind is maybe like kind of like a warrior, the strongest individual, but it's actually the individual who's best adapted to the current climate. That is what survival of the fittest actually means. Okay, So here is an example of how Darwin kind of began to come to this theory of natural selection.
What are Darwin's finches, and why are they so important to biologists?
Great accent.
The study of finches led to the development of one of the most important scientific theories of all time. But how did this come about? In December 1831, a naturalist called Charles Darwin boarded the HMS Beagle bound on a surveying voyage to South America. Whilst the ship and crew carried out coastline surveys, Darwin was free to explore the islands en route. In 1835, the Beagle arrived at the Galapagos Islands near Ecuador. What Darwin found there surprised him greatly. As well as giant tortoises and marine iguanas, Darwin collected and preserved a variety of different songbirds called finches. Upon returning to the UK, he examined them together with ornithologist John Gould and made some fascinating discoveries. The scientists observed that the birds were all similar to a single type of finch found on mainland South America, suggesting that these mainland finches had originally colonized the island. However, the Galapagos finches were all slightly different from the original mainland species, and they were also different from each other. The finches on each island showed distinct variations in their overall size, beak shape, and claw size. These differences were attributed to the differing food sources available on the various islands of the Galapagos. Some of the birds had long, thin beaks and sharp claws suited to catching and eating insects, while others had large, powerful beaks suitable for cracking open nuts. Because of the distances between the islands, breeding between different species of finch was unlikely, and Darwin concluded that the finches must have evolved over time from the original mainland species to suit the conditions found on each individual island. In all, 13 of the birds brought back by Darwin were identified as being entirely new species, all similar to each other, but with definite variations from their common ancestor. Darwin proposed that the variations seen both within and between the finch species arose by chance. Variations which gave any individual a competitive advantage made them more likely to survive and therefore reproduce, outcompeting those with less advantageous characteristics. Darwin called this theory natural selection, and he published it in his book On the Origin of Species in 1859. Evolution by natural selection is now widely agreed to be the most accurate theory to explain the origin and diversity of all life on Earth. In this video, you have learned how the finches of the Galapagos Islands led to the development of the theory of evolution by natural selection. Is evolution of these birds still happening? See what you can find out.
Alright, so to recap that video, Darwin's finches basically, he noticed that there were adaptations that related to their resources. And so subsequently, researchers have actually kind of wanted to, kind of confirm these findings. And so doing this, we can actually observe evolution within these populations in real time. And so in different years, food availability will make certain beaks more favorable, basically. This can be based on drought conditions and the availability and thickness of certain seeds and things of that nature. And actually the traits of offspring in the F1 and F2 generations reflect the resources that were available to their parents. And so parents that had beaks more suited to the available resources, those traits will actually become more common in future generations, which is really cool that we can kind of observe this evolution in real time. And so another example of a way that we can observe evolution in real time is in microbial evolution. So you've probably heard of antibiotic-resistant bacteria or drug-resistant viruses. Like you guys probably know that HIV is known to be very adept at evolving within a human in order to resist different drug combinations that we use to treat it. So microbial populations actually reproduce very quickly, and therefore evolution can occur very rapidly in these populations. And it's important to note that mutations do not arise in an individual in response to a selective pressure, if that makes sense. And so in other words, if there weren't any drug-resistant bacteria within a population to begin with, if we treat them with a drug, they're not going to evolve due to the presence of that drug. The variation has to exist prior to the selective pressure. And so mutations must be pre-existing within the population such that the selective pressure can act on them. So mutations will not arise because of the selective pressure. Instead, they pre-exist, and then they become more common in the population as natural selection acts on them. So basically, if we treat an individual that has an infection with an antibiotic and none of the bacteria within that population are drug resistant to begin with, then we're going to be able to treat that bacterial disease, right? We're going to be able to kill off all the bacteria. But in some cases, and all it really takes is one bacterial organism within the population to be drug resistant, to have this naturally occurring mutation. And as all the others are killed off and that one bacteria, that one individual divides, the population rapidly becomes entirely drug resistant. And so that's another way that we can observe evolution in real time. So some other examples of evidence of evolution within today's world, and there are a lot of them, but for example, genetic similarity is some evidence of evolution. So unless organisms evolved from a shared common ancestor, the question is, why would their genetic profiles be so similar despite such drastic phenotypic differences? And so to demonstrate this, between any two human individuals, they're going to be about 99.9% genetically similar. And that makes sense, right? We all look a lot alike, pretty much for the most part, act a lot alike. And so it makes sense that we share a lot of our genetic information. Humans and chimps, again, fairly similar. They're thought to be our closest living relative. And humans and chimps are about 98.7% genetically similar. So still, remarkably similar. And then if we look at humans and mice, which now we're looking at organisms that, on the surface level look pretty much completely different. And yet we're still about 97.5%. 97.5% of our DNA is shared between us and mice. And so the question is, why would this make sense unless we shared a common ancestor, some amount of generations back with mice way back in the mammalian family? And there's really no other explanation for it other than that basically we share these traits because we evolved from a similar common ancestor. Additionally, we can look at fossil records for evidence of evolution. And so the idea of a missing link, you may have heard this between humans and apes, but this is in itself is a bit of a misconception. And so there are actually many missing links in the documented fossil record that demonstrate the evolutionary path between us and our last common ancestors with apes like chimps. And so there are actually many sort of intermediate steps within the fossil records of basically human-like ancestors that were more similar in many ways to chimps. And so we can basically use carbon dating to figure out when these individuals were alive and kind of trace our evolutionary history in that way. And so there is no real missing link. There are many links along the way, and while we probably haven't uncovered all of them, but we do have a number of links within the fossil record. Okay, so that's it for evidence of evolution. So for today's assignment, go to Moodle and check out the pocket mice video. And so watch that YouTube video. It's not too long on Moodle. And then fill out the questions that are under the Class 1 folder and get that back to me by the end of the day. All right, see you guys Thursday.